Welcome. I'm Rick Carney, Coordinator for the Great Basin Landscape Conservation Cooperative. For those of you who may be new to the LCC, we are a partnership among public and private groups that are working together to meet large-scale conservation challenges across the 5-state Great Basin region. We have supported a variety of conservation projects on high-priority research topics. This series highlights a few of those projects. We hope that these webinars can provide a platform for discussion about the research and its application to the Great Basin. We will keep today's presentations relatively short, about 30 minutes or so, and reserve the second half of the hour for questions and discussion with the researcher. We will read out your questions for the speaker at the end of the presentation. Today's webinar will be recorded and available on our website. On your screen, you'll notice a small collapsed control panel in the upper right-hand corner. By clicking on the small orange arrow, you can expand that control panel. And in this expanded control panel, you can send in your questions. Simply type your question into the chat area near the bottom and press send. The question will be sent to us to respond to at the end of the webinar. I would now like to introduce today's speaker, Dr. Marjorie Matocq from the University of Nevada, Reno. Dr. Matocq is a professor in the Department of Natural Resources and Environmental Science. Her research focuses on studying the geographic patterns of population genetic structure and the processes that underlie such patterns. Dr. Matocq will be joined by John Kasbohm from the Sheldon-Hart Mountain National Wildlife Refuge Complex. John is the Refuge Complex project leader and based out of Lakeview, Oregon. With that, I'd like to turn the controls over to Dr. Matocq.
Okay, thank you very much. Can everyone see my first slide there? Okay. So thank you very much for this opportunity. Even though I am going to be the one speaking during this presentation primarily, I'd like you to know that we've got our whole almost our whole UNR group here in my office to help with questions afterwards. We have Kevin Shoemaker, who is our population demographer on the project. Miranda Krall is the graduate student. Tom Dilt and Peter Weisberg are focusing on the landscape ecology analyses associated with the work, and Evelina LaRusia has given a lot of expertise to the project in a variety of ways. And John Kasbohm is joining us from the Sheldon-Hart Mountain Refuge Complex and will also be available for questions afterwards. So today what I'm going to be showing you is, let's see, these aren't, oops, for some reason this isn't advancing. Okay. Today what I'm going to be sharing with you is our first year results for what is a 4-year project that's being made possible by a number of funding and science partners. And so the science partners listed here at the top, providing a lot of generous funding for various aspects of this project. And also here I'm listing a number of individuals that have been extremely helpful with a lot of the on-the-ground logistics. So we have a lot of people to thank for making the project possible. So now that I've introduced you to the team, let me introduce you to the critter du jour, the very charismatic pygmy rabbit. So you may know that these are the smallest North American leporids. They weigh in at about 400 grams. They typically dig their own burrows or improve those left by others. And they are considered to be sagebrush dietary specialists because sagebrush makes up about 50% of their diet in the summer, even when other high-quality forage is available, and up to 99% of their diet in winter is made up of sagebrush. What we already know from the literature and what we have observed in the field ourselves is that pygmy rabbits do tend to be found in areas with tall, dense sagebrush in deep soils with a fairly open understory. They're found throughout the sagebrush ecosystems of the Intermountain West, and the Columbia Basin populations in Washington were identified as a distinct population segment and listed as federally endangered in 2003. But federal listing for the entire range of pygmy rabbits was most recently considered not warranted in a 2010 ruling. So much of our project is motivated by research needs identified in Nevada's Wildlife Action Plan, including first to develop a survey, inventory, and occupancy trend program for pygmy rabbit, determine thresholds for habitat requirements, and gain a better understanding of dispersal ability and connectivity among habitat areas. So in order to begin addressing these, it was clear to us that we needed to begin integrating studies across various spatial scales. So our work is focusing first at the broadest spatial or geographic scale that we're working with here throughout Nevada, southern Oregon, and some into California. Then at a more regional scale, we've chosen 3 areas for more intensive study, and that is the Hart-Sheldon complex in the northwest, the Austin region in central Nevada, and the Elko region in northeastern Nevada. Then within each of these regions, we've identified 2 to 3 localities that will be our finest scale of study, and we refer to those as our demographic sites. So our goals at each of these spatial scales is first at the broadest scale we intend to develop a habitat suitability model and also to identify the geographic distribution of genetic variation throughout that broad range. At a more regional scale, we're conducting occupancy and abundance surveys and identifying the habitat characteristics of occupied and non-occupied sites, and finally quantifying genetic connectivity among localities within each of our 3 regions. Then at the finest scale, within those demographic sites, we are quantifying individual survival and reproductive success and examining local scale dispersal and gene flow. And overall, we mean to capture a gradient of habitats on which these animals rely from here what we think is probably the very good to what they're managing to do in even the bad and the ugly. So today I'm going to take you through some of our progress and approaches at each of these scales of analysis, and I am going to start here with quantifying genetic variation across the broadest spatial scale of our work. And so for this, in collaboration with Eveline LaRusia, we obtained about 200 samples from throughout the range in Nevada and the Mono Basin of California. And we genotyped these individuals at 13 microsatellite loci designed for this species by Janet Racklow's group in Idaho. And using a Bayesian clustering approach implemented in the program structure, we find very strong support for a K of 2, or that is 2 highly differentiated genetic groups. Spatially, those 2 groups fall out like this, where we have the Mono Basin of California being a highly differentiated genetic group from the remaining Great Basin populations that we've included in our analysis. These groups are differentiated by an FST of 0.22, so again supporting that the Mono Basin populations are genetically distinct from the populations in the more main portion of the Great Basin that we've included here. To give you a comparison, the average pairwise FST between populations in the Mono Basin is 0.08, and that's the same for average pairwise differentiation between groups in or populations in our main Great Basin area, again an FST of 0.08. So this 0.22 is really dramatically higher. We know that the Mono Basin populations are approximately 100 miles from the nearest known populations in Nevada, and our genetic data suggests that this spatial isolation is indeed having genetic consequences. So it's likely that this isolation began long ago and has probably continued with Holocene warming and more recent changes to the landscape. Now, if we remove the highly differentiated group of the Mono Basin from our analysis, we discover more subtle genetic differentiation across our Great Basin populations. But these are minimally divergent genetic clusters, and I'm showing them here with 4 different colors. And you can see that there's broad spatial overlap among these groups. Okay. This level of spatial overlap of groups suggests historic genetic connectivity and mixing across the range. Now those are the patterns of differentiation between these 2 major groups that we have in our study region. How do these populations compare in terms of genetic diversity? So the 2 most common measures of genetic variation are the average number of alleles maintained per locus and the average heterozygosity maintained by populations, or that is the portion of individuals in the population that carry 2 different alleles at a locus. And so here in this graph we're showing all of our sampled populations. Along the bottom of the graph here I'm showing the average number of alleles maintained at a locus, and the right-hand axis is average heterozygosity. So the Mono Basin populations on average are maintaining 3.5 alleles per locus and a heterozygosity of 0.55, and that's in comparison to 5.2 alleles on average being maintained per locus in our Great Basin populations and heterozygosity of 0.71. So while the Mono Basin populations aren't dramatically low in diversity, they are statistically significantly less diverse than our Great Basin populations. And to give you a little bit more comparison, the Columbia Basin population studied by Warheit in 2001 had an average heterozygosity of 0.4, while Janet Racklow's very well-studied populations of pygmy rabbits in the Lemhi Valley of Idaho have an average heterozygosity of 0.75, quite similar to our sort of main Great Basin populations. Despite their level of differentiation, though, I will note that the Mono Basin populations do not maintain any unique microsatellite alleles. They only have alleles found in the other Great Basin populations that we've sampled. So here is a view again of their differentiation from the Great Basin populations using a principal coordinates analysis. But again, this level of divergence is entirely due to frequency differences between shared alleles and not necessarily novel alleles maintained in the Mono Basin. We've also quantified variation at the mitochondrial control region, and this is just 340 base pairs of control region data. And I've included here a number of sequences published by S.D. Zumpf in 2010 from animals collected in Idaho. And what we can see is that all pygmy rabbits are quite minimally diverged from one another at this locus, but this analysis does support our previous conclusions that there were likely broad historic connectivities across the range because we find the same or very similar haplotypes spanning much of the range. So for example, here we have a sequence from Idaho, and very, very similar sequences are found everywhere from Via, the Santa Rosas, all the way down to the Steptoe Valley. So very broadly distributed, closely related haplotypes. We've only sequenced a small number of Mono Basin individuals here, but thus far they all share the same mitochondrial haplotype, and so far we've only found that haplotype in one other individual from the southern end of their range in Nevada here in Belmont. So to give a quick summary of the genetics then, The Mono Basin populations are genetically distinct from the remainder of the range that we've sampled, and these populations maintain lower genetic variation than most other Great Basin populations. Great Basin populations of pygmy rabbits are characterized by 4 minimally divergent genetic groups that overlap spatially, and combined the genetic data suggests historic connectivity across the sampled range. And our ongoing work in genetics is to build regional and local data sets, and also to expand to a more genome-wide data set using SNPs or single nucleotide polymorphisms. Okay, so now I'll move back to our work and sort of our overall framework here of project goals, and I'm going to focus down into our individual study regions to show how we're conducting occupancy and abundance surveys and how we're using these data to generate regional scale models of habitat suitability. So our occupancy surveys are based on sort of a tiered approach where we begin at a particular location with what we call a rapid assessment. And this is a systematic 20-minute survey to see if we can find any burrows. If within those 20 minutes we have not found at least 3 burrows, we consider the site to be unoccupied. But we still conduct a vegetation survey in many of those unoccupied sites because that does inform our habitat suitability model. On the other hand, if within 20 minutes we find at least 3 burrows, we consider the site to be occupied and we proceed to a more time-intensive full burrow density survey, and there too conduct a vegetation analysis that will inform our habitat suitability models. For some of the sites where we've done a burrow density survey that appear to be particularly well occupied, we will invest in more time-intensive trapping and actually marking of individuals, and some of those sites then become our long-term demographic sites. So for the rapid assessments, again, we're doing a systematic 20-minute initial survey. If we find at least 3 burrows, then we conduct a full burrow density survey and attempt to identify the spatial limits of a particular colony. When we are doing our burrow surveys, we're taking note of all sign, and so when you're looking for sign of pygmy rabbits, one of the very important things, of course, is to distinguish them from cottontails that are sympatric with them throughout much of their range. So I'll touch on some of the ways that we do that, and one of them, a very obvious visual difference between them, is that they are substantially different in size, with cottontails being almost twice as big as pygmy rabbits. This is the view that you're likely to get of most pygmy rabbits and cottontails, but this is very convenient because their tails are also very different. So pygmy rabbits have little to no tail, whereas cottontails, as their names would suggest, have a very large, white, fluffy tail. Their ears are also very different, with pygmy rabbits having much furrier and round ears in comparison to cottontails. Another difference is in the size of their fecal pellets. Of course, if their body size is half that of cottontails, pygmy rabbits will have much smaller fecal pellets in terms of their diameter, and that's what you can see here. These are the small pygmy rabbit pellets right in comparison to these much larger cottontail fecal pellets. Okay. Foraging sites are also very evident when we're in a very active pygmy rabbit area, and here Miranda has caught a really nice picture of this where we see this sagebrush that has been browsed and clipped by a pygmy rabbit, and we see evidence of that here. So then at both occupied and unoccupied sites, we conduct vegetation surveys, and those entail laying out 2 50-meter transects per site, and we do line point intercept every 1 meter. We're sampling the plants at every 1 meter along those lines, and also assessing shrub biomass and structure. So we only do vegetation surveys in the summer, but we may do occupancy and burrow density surveys in winter, which is also the perfect time to collect genetic samples in the form of fecal pellets, very well preserved on the snow. And so when there is snow on the ground, we can often find these very clear groupings of fecal pellets at the entrance of burrows. And so we can collect 4 to 5 of those pellets, and we try to get approximately 25 samples per site representing individual pygmy rabbits or burrow systems. So in terms of the burrow and vegetation surveys that we were able to complete in our first season, I'm going to go through each of our regional sites. So this is a map of our study area in the Hart-Sheldon region, and this is up here in the northeast of our overall study area. And we were able to conduct surveys at 39 sites, and we found 23 of them to be occupied. And although we are establishing additional demographic sites, the one demographic site that we were able to fully establish this first season is what we refer to as the West Gulch site. For Elko, we surveyed 80 sites and found 29 of them to be occupied, and we were able to establish 2 demographic sites here in near Jigs and what we refer to as the South Fork site. For Austin, we found 19, we examined 19 sites and found 9 of them to be occupied. And again, we're establishing additional demographic sites, but the Indian Valley site is the one that we were able to establish this first season. So with the occupancy information that we have obtained from those various sites within regions, we are starting to generate preliminary habitat models for each of the regions, and I'm going to go through a couple of those right now. Thus far, we've evaluated 31 environmental covariates that include a wide range of topographic, vegetation, substrate, and hydrographic conditions. And what we refer to here as our preliminary model was built using presence background modeling using random forest as the statistical model. Here on the left, we're showing the existing best habitat model by the USGS GAP program. And as you can see, it shows the vast majority of the Hart-Sheldon region as suitable. In contrast, our preliminary regional model, calibrated with local data, is much more specific and suggests that only 13% of the study area may be high-quality habitat. For Hart-Sheldon, the model performed well in terms of AUC and accuracy. The most important predictor, accounting for 27% of variable importance, was height above the nearest drainage, and this constrained pygmy rabbit occurrence to within approximately 90 meters of drainages, with probability of occurrence dropping dramatically with distance from drainage. Pygmy rabbit occurrence was also associated with high herbaceous cover, modeled as a threshold effect, with occurrence likely only when herbaceous cover exceeds about 20%. Habitats modeled as more suitable also had greater NDVI, or greenness values, indicating more dense, tall sagebrush with a low amount of bare soil interspace, with slight declines in suitability for areas with the highest NDVI values, indicating areas probably with some juniper cover. Finally, habitats were modeled as unsuitable that had high flow drop, reflecting the affinity of pygmy rabbits for relatively flat sites near drainages. Using the same approach for our Elko region, we see again that the GAP model predicts a lot of suitable habitat, whereas our preliminary model, driven by local data, suggests that only about 24% of the study area is quality habitat. The Elko model did not perform quite as well as the Hart-Sheldon model, but also found support for ecologically sensible variables for pygmy rabbit, with the most important variables including NDVI, the topographic position index, and also flow drop and sagebrush cover. There was a distinct peak in NDVI around 0.25, probably associated with the average NDVI of sagebrush. The coarse scale topographic position index showed a preference for lower valley bottoms over mountains, and the flow drop showed a peak around 2 degrees with a precipitous drop for slopes of greater than 10 degrees. And then pygmy rabbit also had a strong preference for sagebrush cover of greater than 10%, although at least at this stage it tended to plateau out much beyond 20%. So as our abundance, occupancy, and vegetation surveys continue in the coming years, obviously these habitat models will become more and more refined. So back to our framework, I'll bring us down now for the end of the presentation down to our finest scale and show you the progress that we've made there. So the sites, the demographic sites within regions that I'll be focusing on are here, the West Gulch site in the Hart-Sheldon region, for the Elko region, the Jig site, and for the Austin region, the Indian Valley site. And again here, when we find an area that seems to be heavily occupied, we actually invest in the very time-intensive effort of trapping and marking these individuals. And we start out by finding the most heavily used burrow systems, so lots of signs. We're looking for lots of fecal pellets, crop marks on the sagebrush, and active burrows. And we set up to 10 traps per burrow system based on this level of activity. We have at least one inward-facing burrow trap per burrow system and outward-facing traps. We also find good runways within about 20 meters of active burrow systems to place traps in those well-used runways. And we always cover the traps with burlap and dirt and other things to disguise them. We place about 100 traps per trap session and run that set of 100 traps for approximately 4 nights. And in the spring, when temperatures are still cool, we can do both early morning and evening trapping. But in summer, we tend to just do a night and early morning session to avoid the hotter evening temperatures. Once we have animals in hand, we're taking a variety of morphometric measurements. We take an ear biopsy for a genetic sample and insert a pit tag for long-term individual identification. So to evaluate the population density and in the future to quantify demographic rates like survival, we're using a spatially explicit capture-recapture framework called Seeker. And in Seeker, the activity centers of all individuals are modeled explicitly, and detection probability is modeled as a function of the linear distance from the trap location to the activity center. So here in this figure, for the West Gulch site at Hart-Sheldon, you can see that pygmy rabbit activity within the trapping period, again that 4-day period, is generally confined to within about 80 meters or so from their activity center. Seeker is very well suited for estimating density for species like pygmy rabbits that first maintain spatially distinct home ranges such that the degree of exposure to the trap array for an individual varies across individuals. Seeker is also a very good model for animals like pygmy rabbits that persist in colonies that may extend beyond the spatial extent of our trap array. So our 95% credible intervals at this point are still quite wide because we've only got 1 year of data in these estimates, but as we collect more data, we'll be able to estimate all of the parameters, including density, much more precisely. Here are the probability of capture data for the Jig site within our Elko region, and you can see that the relationship between capture probability and distance looks very similar at Jigs. to the previous site that we just showed, which is beginning to suggest this may be a trait shared broadly among pygmy rabbit populations. Now at our Indian Valley site in the Austin region, it turns out that pygmy rabbits were generally captured much nearer to their putative activity centers, and as we'll show, this site is also characterized with seemingly very high densities. So this figure illustrates the posterior distribution for pygmy rabbit density at one of our sites in the Hart-Sheldon region. It's based on several sessions worth of data representing several weeks, and we are able to generate a fairly precise estimate of pygmy rabbit density. So here we are estimating 2 pygmy rabbits per hectare with a 9 to 5 probability that our density falls within 1.4 to 2.7 individuals per hectare. And again, this is sort of our first step towards our ultimate goal of fitting a model of regional densities and abundance dynamics for this species. For the Elko region, the Jig site appears to have slightly higher density than the West Gulch site, both of them being well within the range of what's been reported for other pygmy rabbit populations though. So here we're estimating almost 7 individuals per hectare with a 95 probability of the density falling somewhere between 5 and 9 individuals per hectare. Now again, for our Indian Valley site, we have an extremely high estimate of density with 30 individuals per hectare. And we're still trying to figure out exactly why this area is so densely occupied, but it does stand out as an anomaly not only in the populations that we're studying, but also in what's been previously reported in the literature. So here is the fine scale distribution of individuals, just comparing our West Gulch and Indian Valley sites. We can see the number of individuals of adult females, adult male, juvenile female, juvenile male that we were able to mark and sample this first season. And you can see just even from these raw data with the difference in the distance measure here that the Austin site is extremely high density. So we have some to learn about why that would be. So now that we have these demographic sites established, though, we'll be able to follow these individuals and their progeny through at least 3 more years, and coupled with genetic data, we'll gain insight into the spatial scale of movements and essentially the size and dynamics of these pygmy rabbit neighborhoods across various habitat types. So as a final summary and to sort of give you the places that we're headed in the coming years, in the end we hope to have broad and regional scale habitat suitability models based on our habitat correlates of abundance. We hope to identify the habitat and environmental factors that facilitate or impede gene flow among populations. And then at a fine scale, again, examining patterns of survival, reproductive success, and fine-scale dispersal in various habitat types. And with all of that information, we hope to ultimately develop individual-based process modeling tools so that managers can begin to explore the impact of various types of disturbances or other environmental or climate shifts on the local and regional distribution of pygmy rabbits, and further to understand the genetic consequences of those types of changes. So I'm going to end there and say that with several years left in this project, we are definitely looking to expand our collaborative network. So I encourage you to contact us if you're interested in helping us learn more about pygmy rabbits. And we're interested in hearing from you if you know of particularly active sites occupied by pygmy rabbits. I'd be happy to hear from you if you could lend us a hand in collecting fecal pellets in winter, all the way to if you happen to know if there are other funding opportunities out there that might help us expand some the science outcomes of the projects. We're very happy to hear from anybody on all of those fronts or with any other questions. So thanks so much for your time in listening. We look forward to your questions, and thanks very much for your interest in these animals.
Thank you, Dr. Matocq, for that presentation. It was very interesting. We already have a handful of questions that have come in through the chat box. But a reminder to our audience, if you have a question for our speaker today, please go ahead now and enter that into your chat box. That can be accessed on the control panel for the webinar software. You may need to click on the red arrow to expand your control panel so that you can get to the box to enter your question. But to start us off, we've got a question about the habitat surveys. So you mentioned the use of vegetation survey for assessing habitat. Were there any discussions in including soil surface characteristics such as soil surface texture or rock fragment content in your habitat assessment?
Okay, so again, this is Marjorie, and for various questions, we'll sort of have different folks chime in that have more expertise on the topic. So right now, Tom Diltz is going to address that since he's doing most of our habitat modeling.
Okay, so for our habitat models, we used a combination of Surgo, which is the USDA soil database, and StatsCo data for these regional habitat models. But we are also collecting data at the sites as we do our vegetation surveys on soil texture, rock fragments, and depth. That answer the question?
I just actually received a follow-up from that individual, and so I'll just read that here as well. Put more broadly, my question about soil surface characteristics in pygmy rabbit habitats is, what was your process for determining the types of characteristics to study in the pygmy rabbit habitat assessment?
Well, a lot of it was just based on what we ascertained from the literature, and so we wanted to at least capture those items that we thought had been found to be important elsewhere, but then to expand those some so that we had an opportunity to discover perhaps new variables that hadn't been as important in other parts of the range that have been more well studied.
Great, thank you. We will move on to another question here, and if, again, if anyone has questions that don't fully get answered, there is contact information on the screen for Marjorie. Please feel free to continue to follow up with her if you'd like to further discuss any of these topics. But the next question on here is related to the occupancy survey sites. So how did you select your sites where you surveyed for pygmy rabbits? Was it random or based on preferred habitat characteristics for pygmy rabbits?
I'll jump in.
Okay.
It was a combination of all of the above. So we had random sites, especially for Elko. We had repeat surveys of sites where we had previous indication that there was pygmy rabbits. And we also did some very preliminary habitat modeling with Landsat imagery to identify areas that might be suitable, and we went to those areas as well. So it was a combination.
Okay, great. And kind of to follow up with that a bit, looking at some of these presence or non-presence surveys, in using a 3-borough criteria to determine presence or non-presence, how did you address areas that showed other presence indicators, such as pellet runs that don't seem to lead to burrows?
Well, we sort of assessed at the time if there were a lot of indicators, like for example, if we saw like 3 pygmy rabbits at a site, we would do a little bit more intensive of a survey in the surrounding areas to make sure that we didn't miss anything. But if everything indicated that there was no presence there and we weren't finding any active burrows, then we would just leave it at the 20-minute survey.
Great, thank you. And here another question about the detectability. So given the drop-off of detectability with distance from burrows, over what spatial area are the density estimates applied to?
Well, in the seeker framework, we're trying to extrapolate to a larger area than we're trapping, but essentially, you know, we're trying to get the overall density within that area. Of course, it can change outside that, and you know, so we can't extrapolate too far beyond that. But it of course applies fairly locally, and then we can start looking at covariates that might influence those density estimates and extrapolate using a model using covariates to potentially extrapolate much more broadly than these estimates are capable of.
Thank you. I will remind the audience, if you have any questions for our research team here, please go ahead and enter those now into your question box on your control panel. That was the last question that I had so far, so I would like to give the audience just another minute or two to add in any additional questions that they may have. And while we're waiting for any additional questions, I'm going to switch over to my screen just to show a slide with some additional information on the webinar series. Okay, and we've had a few more questions come in, so I'll go ahead and pose these to our group. Do you have any speculations as to why the Austin site has such high densities?
We're going to let Miranda, our graduate student, speculate.
My initial thoughts are that really, like, the patch size, we kind of expanded and kept searching further than where we trapped, and we didn't really find a lot else. So I think it's just that it's good habitat, but the patch size is pretty small for what they're used to. And that's just my thoughts. That is not based in anything other than that.
Great, thank you. And any thoughts on the impact of horses or other species on the pygmy rabbits?
We're all looking at each other to see if someone else has expertise. Maybe John from Hart Sheldon could take this one, because that is such a great study area to be thinking about potentially the impacts of those types of animals on the landscape in general, or these animals in particular.
Well, I can address that a little bit. I think I'd have the very same question. We just removed all the horses from Sheldon, and seeing how that population responds would be a big issue. Horses have pretty big impacts on anywhere near water, so I'm guessing there might well be a negative impact of horses in that area, but I couldn't quantify what that is.
Thank you both. Another question here is, has anyone extrapolated the impacts of climate change on the species, meaning the pygmy rabbit? And then related, what is their home range size?
Well, their home range size, that varies a lot. depending on the habitat. So I would actually, if I could follow up with that one later, just to talk about sort of the range of home range sizes that have been found in different habitat types, because I otherwise wouldn't have those numbers right off the top of my head. In terms of extrapolating the impacts of climate change, that's exactly what we hope to do with our process modeling tool that we'll develop, so that those would be the types of models that we would put in all of the parameters that we're learning about now in terms of individual survival, reproductive success, mating system, and dispersal capabilities, and actually model that across different types of landscapes and versions of habitat suitability, essentially, into the future.
All right, thank you. I think the horse question has spurred a few other questions about impacts to the pygmy rabbit. So I'll just group both of these together. The first one is, are the rabbits sensitive to noise from traffic and industrial use? And then what about any observed cattle or sheep grazing impacts?
I mean, so we have the South Fork site especially is right along a main highway through Elko, and I mean, we saw a couple hit on the road that were dead, but it didn't really seem to impact them at all. Like, they were really dense along the road on either side.
And it may be that the loose soils that are associated with those kinds of sort of man-made disturbances actually facilitate burrowing, and so we do see some occupancy right along roads. In terms of other forms of grazing, we just haven't been able to look at that ourselves. I don't know if John would have more to say on that.
I think that might be something that could be done with this data, depending on the individual grazing going on these different areas. For our refuges, we don't have horses or cattle, so that might be a point of comparison that you might be able to work into your modeling efforts.
Absolutely.
Great, thank you all. I'll just one more time ask for any final questions from our audience. We have answered all the ones that have come in so far today, so just give another minute for any final last questions. Go ahead and type those in and send them my way. All right, seeing no additional questions come in, I will just remind everyone that there is contact information here on the screen for Marjorie and John, and this presentation is also recorded, and the slides as well as that recording are going to be viewable later this week. Oh, one more final question just popped in right as I was saying that, so I'll take this one here, and that is, any speculation about the one site that had a significant difference between adult males and females, 13 versus 25?
It's just too early to tell at this point. Yeah, it's just too early to tell. We don't. We just haven't been able to spend enough time with these animals to figure that out. The males are both, whether at the juvenile stage or adult stage, that West Gulch population has proportionately fewer males than females, and that's just going to be something that we have to study through time. It could just be an anomaly of this first season, but we'll have to track that through time.
I have an additional,
okay, thank you.
Just quickly.
Oh.
We trapped West Gulch during June and July, which is kind of the big part of the breeding season. And males have been shown to have larger home ranges than females in breeding season. Whereas for Indian Valley, we trapped it more towards the beginning of August when maybe males weren't traveling as far out, their home ranges weren't as big. So that could maybe have something to do with it.
Great. One more question just came in here. So how close in proximity to cottontail burrows have you found pygmy rabbit burrows in Nevada?
So cottontails don't actually make burrows like pygmy rabbits do. They have been shown to use pygmy rabbit burrows, and I actually have a video in Idaho of a pygmy rabbit entering a burrow and a cottontail entering a burrow a little while later. So they do use the same burrows, but their cottontails don't actually make their own burrows.
Great, thank you. All right, at this point, I believe we've gotten through all of the questions from the audience. So I'm going to ask, Linda, are you on the line?
I am.
All right, I'd like to turn it over to Linda Allison, who is the Interim Science Coordinator for the Great Basin LCC, to say some closing words here.
All right, so on behalf of Rick, I'll just say thank you for attending today's webinar. A recording of the presentation will be available on our website later this week. If you have any questions about the LCC or this research, please feel free to contact Rick, Marjorie, or John. Their contact information is displayed on the screen. Today's webinar was the final presentation of our 2016 series. Join us this January for a virtual public forum where you can suggest topics for the 2017 webinar series. More information will be available on our website very soon. And last but not least, we really want to know what you thought of today's webinar. Please take our short 2-minute survey that will appear on your screen as you log off. Your feedback is really valuable, and it helps us plan and improve future webinars. Thank you.